In this podcast, I'd like to talk about germline mosaicism, and then we'll spend some time talking about the concept of penetrance. Germline mosaicism is an interesting phenomenon. It could be the cause of an autosomal dominant condition or an X-linked condition when there's no family history of the disease. And this can be quite puzzling at first, because if it's an autosomal dominant condition, the spontaneous mutation rate for diseases is relatively low, but no family history, and suddenly they have a child with an autosomal dominant condition. That may well be a result of germline mosaicism. As we come to appreciate, during embryonic development, a mutation may occur in one cell, and then the daughter cells of that cell are all going to contain the same mutation. And that's what's shown schematically in this diagram. So a mutation occurs in this cell during embryonic development, and then all the daughter cells in the developing organism that result from that cell contain the same mutation. So the mature individual would thus be a mosaic of mutated cells and non-mutated cells. Suppose this mutation occurs in a primordial germ cell of the embryo and not in a somatic cell. That then becomes very interesting from the point of view of genetics, because the person would appear phenotypically normal but could transmit a mutation to their offspring. And this was first discovered with a lethal form of Osteogenesis Imperfecta. OI is caused by a mutation in the type 1 collagen gene, and people first came aware of this when father and several children were studied. PCR did not find the mutation in the father's skin fibroblasts, but it did find a mutation in approximately 12% of his sperm cells. So the phenomenon of germline mosaicism can elevate the recurrence risk for offspring of a mosaic parent. Because here's the situation. Let's say a child is born, no family history, osteogenesis imperfecta. Well, you might counsel that's such a rare occurrence. I don't know exactly what the occurrence rate is. I forget. Let's say it's like 1 in 15,000. So you might say, well, the recurrence risk for that disease for your next child is 1 in 15,000 because you had a spontaneous mutation in the first case. But if a parent is a germline mosaic, then the recurrence risk is quite a bit higher than 1 in 15,000. This is a pedigree that would have explained germline mosaicism in the original case that was studied in Osteogenesis Imperfecta. This male was normal, but a significant percentage of his sperm contained a mutation in the collagen 1A1 gene, and he fathered children by 2 different women, and those children both had osteogenesis imperfecta. Let me talk about reduced penetrance. This is a situation where an individual with a disease-causing genotype may show a normal phenotype but will still transmit the disease. So what I show on this slide is a pedigree from a family with retinoblastoma, and it shows reduced penetrance. Retinoblastoma is an autosomal dominant condition, and on this side of the family tree you see a vertical transmission. Offspring 50% likely of getting the disease. This individual is an obligate carrier and has the same genotype as all of the affected family members, but this disease shows reduced penetrance. The penetrance for retinoblastoma is about 80%. This can cause consternation as you think about counseling. That means if a disease is 80% penetrant, 8 out of 10 individuals with the genotype will show the phenotype. The individual may not show the phenotype, but will still transmit the mutated gene. So in this side of the family tree, it's an autosomal dominant condition, 50% chance of having an affected child, but it's 80% penetrant. So there's a 50% chance times 0.8 that an individual will show the phenotype of the disease. This individual showed the disease, but his sister did not show the disease. The sister had the defective gene and her children had the disease. So it's a difference in reduced penetrance. So what you would say is it's a 50% chance of showing the disease if it's 100% penetrant, but this disease is 80% penetrant, so 50% times 80% would be a 0.4% chance of showing the disease phenotype. So what do we mean by complete penetrance? By complete penetrance, we mean that the gene is 100% penetrant. If you have the mutated gene, 100% of your offspring are going to show the diseased phenotype if the gene is transmitted. If it's incomplete or reduced penetrance, depending on what level of penetrance it is, that level of offspring are going to show the phenotype even though the gene is transmitted. So you can have complete penetrance, you can have relatively high penetrance, say 80% penetrance, or you might have very low penetrance, where you may have an autosomal dominant condition, but it may only be 10% penetrant. Incomplete penetrance can occur in autosomal dominant and autosomal recessive conditions. The BRCA1 gene is an autosomal dominant gene that causes breast cancer. It's 80% penetrant. There are some forms of hemochromatosis that are autosomal recessive and that are only 5 to 10% penetrant. These are just some examples. We also talk about age-dependent penetrance, and this can be observed for some genetic conditions. Huntington disease is an autosomal dominant condition, but it shows up later in life, so we say that it's age-dependent. Here it's just showing that you start seeing penetrance of a disease like Huntington's somewhere in 30s, maybe young 30s, and it's slightly different for when it shows up in males or females. Such diseases are generally not affected by natural selection, and so the gene frequencies remain high. And it's obvious why they're not affected by selection, because before the gene is manifest, individuals have already reproduced. This clearly brings some ethical questions to mind in relation to genetic disease. So, for example, you have a parent that has Huntington disease. Would you want to know that you have a 50% risk of having the disease yourself? You may choose not to know it. On the other hand, if you know you have that 50% risk, do you want to have children and pass the disease on? It also raises an interesting set of issues from the point of view of what do you want an insurance company to know? We do have in our country genetic non-discrimination laws, so it is not technically legal to discriminate against individuals in getting health insurance if you know that those individuals might be carrying an autosomal dominant condition. With the Affordable Care Act, genetic discrimination may not be an issue, but health insurance is still in a state of flux in our country. Let's talk about penetrance and contrast that with variable expression. Penetrance is either an all or none phenomenon. One either has the disease phenotype or you do not have the disease phenotype. You may have the gene, but the phenotype might not be expressed. Variable expression refers to the degree or severity of a disease phenotype. This may be caused by environmental factors, interactions with other genes, or different types of mutations at the same locus. Neurofibromatosis type 1, for example, an autosomal dominant disease. It has variable expression but 100% penetrant. We know that there are some individuals that have neurofibromatosis that has very mild expression. They may have a few cafe au lait spots. They may have a few Lesch nodules or benign growths on the irises. They may have a few neurofibromas, a non-malignant peripheral nerve tumor. On the other hand, Other individuals, even in the same family, may have very severe expression, hundreds or thousands of neurofibromas. They may have optic pathway gliomas. They may have learning disabilities, hypertension. They may even have scoliosis. So this is a situation where there's 100% penetrance, but variable expression. Not so important for you to know right now, the gene that causes neurofibromatosis type 1, NF1, it's a tumor suppressor gene. The product is neurofibromin. As with reduced penetrance, variable expression allows disease alleles to survive at higher frequencies in populations. Pliotrophy is a common feature of human genes, and pliotrophy is a situation where effects are exerted on multiple organ systems. Marfan syndrome, a good example of this. It's an autosomal dominant condition. A mutation in the fibrillin gene results in unusually stretchable connective tissue, and you know from your study of histology that connective tissue forms parts of all organs. Marfan syndrome mostly affects the ocular system, the skeletal system, and the cardiovascular system, but every organ can be affected to one extent or another because every organ has connective tissue. Cystic fibrosis, another example of a disease where the genes cause pliotrophy. Mutations in the cystic fibrosis transmembrane conductance regulate a gene. The gene product, as we've said before, is a chloride ion channel, so you see it in sweat glands, in the lungs, and pancreas. There are a variety of mutations. The most common mutation is a phenylalanine deletion at position 508. It's an autosomal recessive condition, but because there are many different mutations in this CFTR gene, some patients will have mild problems and nearly normal lifespans. Others may have very severe problems and may not live too much longer than their teenage years. Locus heterogeneity is a single disease phenotype that can be caused by mutations at different loci, even genes on different chromosomes. Adult polycystic kidney disease is manifest by progressive accumulation of renal cysts. You also can see cysts in the liver. You see hypertension, cerebral aneurysms, and even some cardiac valve defects. It's an autosomal dominant condition. There are mutations in the polycystic kidney disease 1 gene that's on chromosome 16. There can also be mutations in the polycystic kidney disease 2 gene, which is on chromosome 4.